the Awesome Tutor, and today we're going to be looking at developing new medicines in the Unit 1 topic of GCSE biology. Now, a drug is tested to see if it is effective, if it actually cures the disease. But some medicines, they're not made to cure the disease, some they're just made to ease the symptoms. So a drug is tested to see if it is effective, if it cures the disease, or eases the symptoms. And it's also tested to see if it is safe. What do we mean by safe? Its toxicity, if it's too poisonous to take, or any side effects that might be created with it. We do not want any harmful side effects. And it's also tested to see what is the most appropriate dose to take it in. Now we have something called double-blind trials. Double-blind trials, it involves the doctor and the patient not knowing if the patient is taking the new drug or the placebo. And that makes the question, what is a placebo? A placebo is a substance that has no drug at all. It's usually an inactive or stable substance, so it does not have the new drug being tested. And it's usually used as a control in the clinical trials. But sometimes it does contain a different drug that's already used to treat the disease. And why would they do this? Well, if for the clinical trials, the patients, they basically volunteer. You cannot force someone to take an untested drug, a drug that's not fully tested. So they volunteer. And if they're going to volunteer, what's their incentive for volunteering? If the drug actually works, they are cured. So that's why sometimes the placebo contains a different drug already used to treat the disease. So the patient is not deprived of having the treatment that he or she has volunteered for. And now we're going to move on to the actual steps in the preclinical trials and clinical trials and blah, 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 until the medicine is on the market. Okay, so this is a typical process through which the drug goes through before it is put on the market. And even then, after it's put on the market, it's still monitored. So the preclinical trials. First, the drug is tested on cells, tissues, and animals. And in the preclinical trials, it basically acts as a model to see how the drug would affect us humans. Now, obviously, there are some ethical issues with testing untested drugs on animals, and they might ask you questions on that, or they might even give you a 6-marker, highly unlikely, but they might ask you questions on that. Obviously, unethical to test on animals, because it might kill them, and that's not good. So, as you can see, it takes a long time to test the drug, from 6 to 15 or even 16 years. And why do we test drugs so extensively? Because if there is something wrong with it that we have not found and is on the market, it may have dangerous side effects like thalidomide, which we shall talk about later. So, as I said here, cells and tissues. It's usually done on cells and tissues to see if the drug does its job, or to see if it is toxic or how safe it is. Because obviously, if it damages the cells or tissues or kills them, then obviously it's not going to be safe on humans. And for animals, testing on animals is to see how it works on a whole living organism. Because as I said, it is a model for how the drug will affect us humans. Then in clinical phase 1, before the drug is tested on patients, it's tested on healthy volunteers. Obviously volunteers, because you can't force people to take the drug that has not been fully tested yet. So, healthy volunteers. Why healthy volunteers? To see any possible side effects. And obviously it's given in a low dosage, because if it's given in a high dosage and there are dangerous side effects, you might kill the healthy volunteers, and that's not going to be good. Same thing with the animals. It's to see any possible side effects as well as any possible doses. So it's basically just in the planning phase to see any possible things that could be done to make the drug better. And now onto the clinical phase 2, where it's actually tested on patient volunteers. And this is mainly to see how effective the drug is, if it cures the disease or if it at least eases the symptoms. Then, on to clinical phase 3, where that one drug out of 10,000 versions of it has successfully passed the preclinical, clinical phase 1, clinical phase 2. Now on clinical phase 3, where it's tested on a lot more patient volunteers. And here it's mainly this test for the optimum dosage, because if you take too much of the drug, it may be dangerous. If you take too little, it's not going to do anything. And then it's put on the market, and as I said, it's still monitored to see if anything has been missed in the clinical phase trials. And now on to thalidomide. So thalidomide, why do we need to know about thalidomide? Well, you are required to know about thalidomide in your specification, and I have seen a whole question on it in an exam paper, so it's definitely useful to know stuff about it. Thalidomide was a drug developed in the 1950s, originally as a sleeping pill to help people who could not sleep properly. Then it was later discovered that it could cure or treat morning sickness during pregnancy. But it was not safe for developing fetuses, and the person taking the drug was not actually affected, but the fetus was born with limb deformities. Missing arm, missing leg, blah blah blah, tragically. And then it was recently discovered that it could treat leprosy. But again, children were born with limb deformities. But now, it is discovered that it can treat autoimmune diseases, where the body attacks its own cells. And even blood or bone cancer, it can treat them as well. But it is not used at all now on anyone that's pregnant or could become pregnant. We don't want any more limb deformities. Yeah. Bye.